present today is using the natural resistance of neuronal subpopulation to identify therapeutic targets in neurodegenerative disease, which was presented by Krista Pillar in March 14th, 2014. This paper is almost 300 pages long, so within this 10-minute presentation, I can only cover part of her research. Neurodegenerative diseases are the diseases that are caused by progressive loss of structure or function of neurons. With different diseases, there will be a selective loss of the corresponding neuronal function. Here, the author selects ARS as her main focus. ARS, amyotrophic lateral sclerosis, causes muscle weakness and atrophy throughout the body due to the degeneration of upper and lower motor neurons. So ARS is a disease with selective effects on individual motor poles. The motor neurons in the brain stem and spinal cord degenerate. It's adult onset and the prevalence increases with age. It's more common in men than in women. The patient will have difficulty breathing and swallowing. There's definitely no cure, so new therapeutics targets are needed. Stephen Hawking is one of the famous person who suffers from ARS. This picture shows that some kind of motor neurons are ARS resistant and some are ARS susceptible. Pictures on the left here are cells from control patient. Right are an ARS patient. For ocular motor and all nerve nucleus, the motor neurons are largely preserved, while for lumbar spinal cord, these cells are almost completely lost. Thus, researchers focus on the gene expression of these three subtypes of neurons and want to find which genes are responsible for ARS resistance in different subtypes of neurons, which are ARS susceptible ones. Then they can inhibit the susceptible ones or activate the resistant ones to get a cure for ARS. They use microarray profiling of isolated wild-type motor neurons to identify a gene expression of ARS resistant and ARS susceptible motor neurons and found 19 such genes. The green portion here represents the common area shared by two subtypes of ARS resistant neurons, and the red portion is the area causes the ARS disease. Within these genes, the author used MMP9 matrix metalloprotease 9. It's a potential disease susceptibility genes. She used an MMP9 knockout mice to examine the MMP9 expression level in different motor neurons by microarray. The result here showed that first row, which are all extraocular muscles, doesn't have MMP9 positivity. In contrast, the second row, MMP9 is highly expressed in trigeminal and facial nuclei, as well as in L5 and L6 spinal motor neurons. L5 means vulnerable lumbar level 5. So MMP9 is absent from ARS-resistant populations in midbrain and spinal cord, but present in many other motor neurons. So does that mean high MMP9 level is a marker for fast fatigable motor neurons? To answer this question, she did two experiments. First, measure the size of the motor neurons. There are actually 3 subtypes related to ARS. 2 of them, FF and FR, are bigger than and more vulnerable than the other one, S. By comparing the size of MMP9 high or negative motor neurons, she found that the largest is the MMP9 positive, and the smallest is the MMP9 negative. And the second experiment is to inject chlorotoxin into muscle. It will be retrogradely transported of axons and label the cell body. So in this way, she can identify a specific motor pool that connects to the specific muscle. Now she knew that tubular anterior muscle, which is TA muscle, is primarily made up of fast twitch fibers, and soleus muscle is made up of slow fibers. Then she stained these muscles with MMP9. The hypothesis is that fast muscle, which is TA muscles, would have more MMP9, and slow muscle, which is soleus muscles, would have less MMP9. The result is consistent with the hypothesis. Soleus muscle, we can see, has much more MMP9 negative muscles. Thus, she proved that high MMP9 is labeling the fast fatigable motor neurons. And next, she wants to prove that MMP9 is a marker for vulnerable motor neurons in ARS. To test this, she used an animal model of ARS, SOD1 mice. She compared the percentage of motor neurons lost at the end stage to the percentage that expressed MMP9 in wild-type mice. The result shows a strong positive correlation, which suggests that motor neurons that express MMP9 are subsequently lost in ARS. Then the author wants to prove that MMP9 is not only a marker for vulnerable motor neurons in ARS, but also actually contributes to motor neuron degeneration in ARS. And so here she used SOD1 G93A mice. It's a mice that had a mutation of superoxide dismutase, where glycine is replaced by an alanine, which is frequently observed in ARS. So these mice have clinical manifestation and exhibit pathology similar to human ARS. They cross MMP9 negative mice and SOD1 G93A mice to make the mice more disease resistant. So in black, this is the mice model with MMP9, shows nearly complete denervation by end stage. The innervation of TA muscle is 50% complete by 33 days in normal SOD1 G93A mice, but delayed by about 80 days by removal of either one or both alleles of MMP9. This shows that denervation of hind limb muscles is delayed in the absence of MMP9. Furthermore, they observed significant improvement when MMP9 ablation in other functional tests, like the swimming test. And then they measured the compound muscle action potentials, CMAP, to confirm that the preserved synapses were functional. She put recording electrodes into TA muscle and then stimulated the sciatic nerve and found that When MAP9 is intact, the CMAP in SOD1 G93A mice was reduced to 10% of that in wild-type controls. However, deletion of 1 or 2 copies of MMP9 rescued up to 50% of normal values. So summary, functional subtypes vary in their disease susceptibility in ARS. FF is ARS susceptible and S is ARS resistant. The author chose a potential disease susceptibility gene MMP9 as a target, and she proved that high-level expression of MMP9 is a marker of ARS susceptible motor neurons, and MMP9 actually contributes to the motor neuron degeneration in ARS. Thank you.